Hi, my name is Riley Hamel, and my research involves the investigation of post-translational modification on NHE1 transport function. The protein that we study in the Provost lab is a sodium hydrogen exchanger isoform 1, or NHE1. This protein is an integral membrane antiporter, meaning that it exchanges one extracellular sodium ion for one intracellular hydrogen ion. It has two main functions in mammalian cells: to regulate pH within the cell and to function as a membrane scaffolding protein to form stress fibers and regulate signaling pathways in the cell. This diagram on the right is the currently accepted model of NHE1. NHE1 is stimulated in cancerous conditions in order to help keep the cells alive. Intracellular pH dysregulation is associated with cell proliferation. Thus, NHE1 is a potential therapeutic target against cancer. NHE1 is phosphorylated by 11 kinases, and there are 14 known and predicted sites. The kinases that we focused on in my experiments were MAPK, AKT, RhoA, and RISC. MAPK, RhoA, and RISC phosphorylate NHE1 to turn on transporter function, while AKT phosphorylates NHE1 to turn off function. In our lab, we found that NHE1 can be reversibly palmitoylated. This is a novel finding. Such modifications are reported to regulate protein trafficking and protein interaction. Palmitoyl-transferases, or PATs, catalyze the lipidation of NHE1. But PATs can be inhibited by 2-bromopalmitate, or 2BP. This shuts off the palmitoyl-transferases, while thioesterases depalmitylate NHE1, therefore decreasing overall palmitylation of the protein. NHE1 is palmitoylated, but we don't know where or how. Thus, we ask the questions: which cysteine residues are palmitoylated on the C-terminal domain, and which PAT initiates palmitylation of NHE1? Targeting palmitylation, like phosphorylation, is also a therapeutic strategy for cancers. Post-translational events increase NHE1 transporter function, and post-translational events increase activity of NHE1 in the presence of stimulating factors. Thus, we hypothesize that palmitylation and phosphorylation coordinate to subtly modify NHE1 activity in a novel rheostat of the cell. To quantify this change in transport activity, we measured the change in pHi of Chinese hamster lung fibroblasts using a fluorescent pH indicator known as BCCF. BCCF fluoresces based on the acidity or alkalinity inside the cells. BCCF has a dual excitation emission spectrum that can be used to calculate ratios of fluorescence that, when plotted on a reference curve of pH values, can be used to determine the intercellular pH of the experimental conditions. Factors that stimulate NHE1 activity are insulin, Fetal Bovine Serum, which is a complex mixture of small molecules and agonists that stimulate many signaling pathways to turn on NHE1, LPA, which activates a G protein coupled receptor pathway, and PMA, which activates a signaling cascade involving protein kinase C to phosphorylate NHE1. This is a graphical representation showing an increase in pHi of CCL39 cells, indicating that NHE1 activity is stimulated with FBS over a period of 900 seconds. As you can see, the pHi increases from 7.35 to 7.45. Insulin, PMA, and LPA also stimulate NHE1 activity. Steady-state pHi assays determine that insulin signals palmitylation and phosphorylation of NHE1. This is done by insulin binding to the insulin receptor, signaling IRS proteins, which then activate signal transduction pathways within the cell. This graph indicates that trend when measuring the change in pHi of fibroblasts. What's interesting is that PI3 kinase and AKT are involved within the same signal transduction pathway, but have different effects on transport activity when inhibited. The assays also indicated that serum signals palmitylation and phosphorylation of NHE1. One or many of the agonists or small molecules present in serum may activate a cascade that activates MAPK, thereby phosphorylating NHE1 and increasing activation. This is indicated by the graph on the right that shows serum stimulation. LPA signals palmitylation of NHE1, which increases transport activity as well. However, due to the pandemic, this pathway needs to be furtherly investigated. This is a summary of the results of the pHi steady-state assays. The rheostat mechanism is supported as insulin, LPA, and FBS are all implicated in kinase cascades that palmitylate and phosphorylate NHE1 in different ways. Future directions include investigating other signal transduction pathways that are involved with the phosphorylation and palmitylation of NHE1, conducting acid load recovery assays to determine the transport kinetics of NHE1, and conducting cell migration assays to determine the effect of palmitylation on NHE1. I'd like to thank the University of San Diego Chemistry and Biochemistry Department, as well as the Biology Department. And I'd like to thank Dr. Joseph Provost in the lab, as well as the Associated Student Government. Thank you.